Amanda, and this is Derek. Like she said, we're from Fort Lewis. We want to talk to you today about how Tamarisk affects arthropod and small mammal communities in our study site in Big Gypsum Valley, Colorado. We've been working on this research for the last 3 years. So this is a map of our study site. We're located in the Dolores River Canyon Wilderness Study Area. It's on the Colorado-Utah border in Big Gypsum Valley. I just want to give you an idea of the topography of our study area. So I'm going to go through a few pictures. It's a really beautiful area. We've spent over 60 days out there, so we're kind of in love with it and a little obsessed with it as well. It's a very arid climate out there. Much like the Moab area, there's all of these high mesas, like this is called the Hat here on the left. There, I guess the right too. And you can see it's just a really beautiful area with the canyons, and then you have the La Salles in the background there. So the lifeblood of this area and of our study sites is the Dolores River, and all of our study sites do occur along the Dolores River. We're below the McPhee Dam, so we are in a very regulated area. It's very sedimented, and sometimes we go out there and it's completely dried up, and sometimes it's flooded, so very irregular and inconsistent with the flow. Something also pretty cool out there is that we have some endemic flora. So this is the Big Gypsum Cateye, and it only exists out in Big Gypsum Valley in the gypsum soils there. This picture on the right is the mound that it exists on, and something that's unfortunate is that it's not protected at all. So as you can see, people have been off-roading up the road, and so that's kind of unfortunate. So now that you get an idea of where our site is at, I'm going to dive right into our study. So we looked at how Tamarisk affects arthropod and small mammal populations, and we do this by comparing Tamarisk to two native counterparts. Our first one is a shrub community with mixed alkyne shrubs, and our second one is a willow community. We have 3 years of arthropod and vegetation data, and then just this last year we added small mammal data. Can you guys hear me? I feel quiet. Okay. So this is a really important study because, as we all know, invasive species cost us a lot of money every year and push out a lot of the native flora and fauna which exist here. In arthropod communities, and the effects of invasive on those communities is extremely understudied. And it's important to understand arthropod communities because they make up such a large majority of the organisms that do exist. There's over 1 million species of arthropods, and they're also important because they make up the bottom of the food chain. So understanding how invasives are affecting our arthropods can tell us how they're affecting members further up in the food chain as well. So the main part of our study is the arthropods. We collect our arthropods through pitfall trapping, and it's basically just a cup dug to ground level and filled with 60% ethanol. So the arthropods, they just walk along the surface and fall in, and you have a ready-to-go specimen. We set up our sites in X formations, and we had 9 traps per an X and then 3 X's per habitat. So we had a total of 27 traps set up for each habitat type. We would set out these traps for 48-hour intervals, and then once that interval was over, we would sift out the arthropods. Then we would take them back to the lab, sort out all of the debris and vegetation, and then identify them to the lowest taxonomic level in our capabilities, and then sort them into these individual vials. So this just shows some of our common lab equipment. We also decided to take vegetation data because obviously differences in the vegetative structure is going to influence our differences that we see in our arthropod structures. So we took soil moisture, ground cover, and canopy cover in each habitat type in the formation you see with the green lines there, and then the X's represent the formations of our pitfall traps. And then just this last year, we wanted to take it a step further, so we added a small mammal component to the study as well. We didn't get quite as much data as we would have liked due to floods and other unforeseen circumstances, but we did get a preliminary study going. And how we set it up is we had 50 traps in our Tamarisk habitat and 50 traps in our willow habitats. We couldn't set them up in our shrub habitat because our tamarisk and shrub habitats are adjacent, so then the home ranges of the mammals would overlap. So we would set the traps at sunset and bait them, and then at sunrise we check them, and we're doing a mark recapture study. So if we did capture mice, they were anesthetized, so it is just knocked out in the picture, ear tagged, and then various measurements were taken. So this shows our research sites from the vantage point in the bottom right-hand corner there. The red is the tamarisk, the green is the shrub, and so as you can see they are adjacent to each other, which is why we could not set up small mammal traps in the shrub. And then further down the Dolores, we have the willow sites in the blue there. So I'm just going to go through our sites a little bit. So our tamarisk sites were obviously dominated by tamarisk, with various grasses as the understory. They're characterized by high soil moisture and high litter as well. So this is just some pictures of our tamarisk. They're very variable as any tamarisk stand. Sometimes they're defoliated, sometimes they're flooded, sometimes they're flowering. And we also have a high prevalence of Tamarix beetle at our sites. We've caught a lot of Tamarix beetle larvae and adults who both feed on the tamarisk. So we have observed some defoliation throughout the years, and this will be influencing our arthropod and small mammal results that we find. And then just this last year, we were really excited to catch our first Tamarix Weevil, which is cool because this is really the first time it's been caught in this general area. So we were pretty excited about that, so throw it in there. Our shrub sites are dominated by various native shrubs, which I'll go through in a second. It has a high abundance of rocks and bare soil, and it has a very low soil moisture and low litter. So as you can see, our shrub site at its height has a lot of low-lying alkyne desert shrubs. So what's dominant there is Arthropod canescens, or four-winged saltbrush, Sarcobatus vermiculatus, or greasewood, Forestiera pubescens, or privet, and an unknown species of Guderisia, or snakeweed. Our last site, or our willow site, is dominated by willow and various reed grasses, and they're kind of in the middle with soil moisture and litter for our three sites. So our sites are dominated by Salix exigua, which is narrow-leaf willow, and we do have a prevalence of Phragmites australis in there too, which is important to consider.
Hello. So as you can see, we spent a lot of days out here, about 65 days in the field. We did 3-day trapping periods. And you spend that much time out in the desert and a lot of crazy stuff is going to happen. We've seen everything from dust storms to floods to fires, and it's been quite the adventure. But the real work began when we took our samples back to the lab, where we had thousands and thousands of arthropods to sort through. Here you can see pictures of our weekly lab meeting. And we spent countless hours going through our samples, sorting them, and getting them into little labeled vials. So you might be wondering how a bunch of undergrads could identify any arthropods, since there's a lot that aren't even known to science yet out there. And a resource I found extremely useful is bugguide.net. And if you haven't checked this website out, I really strongly recommend it. It's fascinating, and you can actually post pictures of your bugs on there, and experts get back to you and can help you identify it. So I found that to be a great resource. So now I'll get into our results a little bit. Here you can see we had 16,000 arthropods from last year, and then in 2012 we had 30,000. We also trapped 58 small mammals. So this kind of gets back to what Amanda was explaining about with our different habitat types. This is actually the data that supports that. We just did point intercept, and you can just see each habitat is kind of made up of different things, which is obviously why we chose the 3 different habitats. Here you can see the canopy percent, and shrub obviously is all below knee height, so it's not going to have any canopy, but tamarisk and willow have not significantly different canopy percent, with tamarisk being a little bit higher. So this is one of the more interesting things we found. We did an NMS ordination on the arthropod data. So each one of these triangles represents each one of our sites on a particular date. And what this does is it bases it off the arthropod composition of each site. And so the two most similar sites it places next to each other, and the two most dissimilar sites it places far apart from each other. And then it graphs everything else in between, basically just giving you an idea of how similar the habitat types are in arthropod composition. And here you can see shrub in the green roots out real nice up to the top. And so it forms its own unique arthropod assemblage of arthropods. And so that's kind of interesting to see shrub grouping out, especially since it's so close to Tamarisk, but yet it's still forming its own unique arthropod composition. You can also see here that Tamarisk and Willow have a lot of overlap. So Tamarisk is forming an arthropod community similar to Willow. That's kind of fascinating. But really, all three of them have areas that also aren't overlapping. So all three of these form unique arthropod communities, but Tamarisk is more similar to Willow, and shrub is least similar to the other two. And here on this side, you can see our really neat native tarantula with the cool orange hairs on his butt. So going back to that, how they have different members of the arthropod community, here's average order abundance per habitat. You can see it's pretty much significantly different between every single order. We got the major ones like mites, spiders, a couple of different springtail orders, bristletails, beetle, fly, and so on. But they're all different. So it's interesting to see that each habitat makes its own unique assemblage of arthropods. So if Tamarisk is making its own unique arthropod community, we might want to ask, is that arthropod community just as healthy and viable as Willow and shrub are? Well, here we can look at some other ecological measurements. We have family richness right here, and this actually doesn't significantly differ between the habitats, so Tamarisk isn't decreasing the family difference or richness. Here we have, I'll go skip over here to abundance, because it's not significantly different either between the habitat types. So all three habitats have a good abundance and richness of arthropods. In the middle here we have Shannon-Weiner diversity index ran at the family level again. And you can see shrub is actually significantly higher, but Tamarisk doesn't differ from Willow significantly. So shrub may be higher, but Tamarisk is still comparable to a native counterpart. I'll just take a minute to go over our small mammal species that we trapped. So for the small mammal study, we caught deer mice. The coolest one we caught was the brush rat. I mean, it's like the size of a guinea pig. And then the most we caught of was the canyon mice, not pictured, but harvest mice and pinyon mice, and we caught those too. So here's our results, and remember, this is all preliminary since our study was cut short due to flooding. And but it's kind of interesting to see that the brush rat, we only caught him in Willow. Well, we caught two of them. And the canyon mice are a little higher in Willow, but not significantly. And then deer mice, pinyon, and harvest mice are all higher than Tamarisk, even though not significantly. Just kind of interesting to see. So that'll lead us to our conclusion, which is Tamarisk forms its own unique arthropod community with similarities to Willow. And Tamarisk arthropod community is not decreased in richness or abundance, but and it's not decreased in Shannon-Weiner family diversity index either when compared just to Willow. And shrub's Shannon-Weiner diversity index was greater than Willow and Tamarisk. And we think that the different vegetation composition is likely the main factor in determining the arthropod composition. Tamarisk small mammal community was just as diverse and abundant as Willow, but again, preliminary, not much backing behind that. Here you can see a couple really cool darkling beetles. These are some of my favorite. This guy actually is pretty rare over here with the cool indentations on his elytra. So what implications does our study have? Well, it's important for land management to know that Tamarisk does support a different variety of arthropods, and that's going to provide different resources to wildlife. These different resources may not be the norm for the natural, but it's better than no Tamarisk, than nothing at all, because bare ground isn't going to support arthropods, but Tamarisk will. So it's better than no resources. So if eradication is necessary, we really need to quickly revegetate the area so that arthropod numbers are restored with native community dynamics. So do I still have some time? Still got a little time here, so I'll go through a couple interesting arthropods. This guy is called a piglet bug. He's one of my favorites. He's got a really big red nose. This is a pretty rare species of Hysteria beetle. These guys feed on dung and carrion. This is actually a relative to the black widow spider, and much rarer and much less poisonous. He's pretty cool. And my favorite all-time arthropod is a Bupresti woodbourne beetle. Spectacularly beautiful, quite remarkable, and they are specialists on the Atroplex salt brush that we were talking about earlier. And here's a relative to the yellow jackets. It's in the same family as yellow jackets. We call them the happy face wasp. You can see my little happy face. I'd like to just thank everyone at Fort Lewis for all the help. We had a lot of different researchers who helped us. Thanks especially to Dr. Deb Kendall for making this project possible. And yeah, any questions? Get back up here, Amanda.
I was wondering, when you collected the arthropods in the Coleoptera, what percentage of those were the Tamarix Weevil?
Oh, that's good. That's a good question. This last year, we hardly captured any adult Tamarix beetles in our pit traps. I mean, we would capture a few every time.
Mm-hmm.
Mostly it'd be the larvae. And this last year, our Tamarix were pretty completely defoliated, so we actually didn't catch too many. In 2012, though, we would have thousands at a time.
Okay. I've seen defoliated Tamarix areas. They actually come down to the ground and start moving around trying to find a green plant. So I would expect to catch more, maybe. But
yeah, I think the defoliation maybe in 2012. So we caught a lot in 2012, and I think the Tamarix started defoliating, and this last year it had already been pretty fully defoliated, and we didn't catch as many larvae because of that.
So one of the aspects of engagement is also stability of communities. And I'm wondering, you kind of alluded to this in the answer to the last question, how much variability did you see among years for your arthropods? And do you have a sense that maybe variability was different in kind of pre-vegetation phase?
Yeah, that's an interesting question. Yeah, so we've done the study since 2011, and our Tamarix stands have changed quite a bit, getting defoliated and growing back a little bit. And, you know, we always seem to capture, or at least depending, it's more season dependent. Seems that we always seem to capture around the same amount of arthropods in the same season, but it's very highly season dependent. So early in the season, we'll get a few hundred per site. And then peak season in like June, we'll get thousands per site. And it seems to be pretty stabilized that way.
And we actually took out our slide that was our factors over time because we thought we didn't have enough time, but abundance was significantly different by year. So we had way more arthropods in 2012 than we did this year. And then richness and diversity did not differ by time. So richness and diversity were still the same results, but abundance differed.
Thank you, everyone.